Today, I really need you guys to talk. Let me know if you have questions, what you need help in. I don't have a set lecture for today. I actually plan on it being review day, but I got the dates a little screwed up in my head because the exam is actually on Friday. Your quiz 3 is today, okay? So you have 3 quizzes. I extended the due dates for quiz 1 and quiz 2, and that's because people were having trouble accessing them in the beginning. So I extended those due dates. They're for quiz 1 and quiz 2, they're due today. So if you're still not having access, come on. If you still don't have access, you need to if you don't have access to Mastering Biology, contact Pearson directly. They've helped a few other students and they've been able to get access codes. You need your initial access code from the school bookstore. That's to access Mastering Biology. So you need to have that. I think most everybody has gotten on and done assignments. So that
You muted yourself.
I muted myself. Okay, so how far back did I mute myself?
Just like 2 seconds ago.
Okay, good. Okay, good. All right. So the people, I don't think there's a lot of people complaining about not getting on anymore, but I just want to say, so you get that access code. If you're having trouble with Mastering Biology itself and you have your access code and it's still not working, I've had a few students contact Pearson directly and they were able to get a new access code. So try those steps if you're having trouble, but you have due date today for your first 3 quizzes. All of your homework and labs are due Friday, June 5th. So I corrected my announcement. I had the wrong due date for the exam. Due date is June 5th. So you still have 2 more days to get prepared for your exam. The exam is available now if people want to take it. It is all multiple choice, which makes it easy to grade, makes it easier on you guys too. So it's all multiple choice. There are 70 questions, but there are a lot of easy questions, so it shouldn't be too bad. And I checked, and there is not a time limit. Pearson estimates it will take about 37 minutes to take the exam. So I think you guys will be able to do it within an hour, probably just fine. You're able to use your books, your notes, whatever you want. But I would still encourage studying ahead of time. And that is because if you're spending too much time in your book trying to find answers, you're going to have a huge amount of time spent on this exam. And so being prepared ahead of time, even though it is open book and open notes, is important. Okay. I'm trying to think about if there's other questions that were about the exam. I don't think so. There were a few questions for the attendance that we had for Zoom yesterday. Some of you did your Zoom attendance, so you got credit for that. We're going to do a Zoom attendance again today. And so as long as you just participate and do that by the end of today, which will be a really quick, like 1-sentence answer, you'll get credit. So I want you to make sure you're getting credit for being here on Zoom and being up to date in class. So maybe let's start with chapter 1, and we're just going to kind of do a review. And I think Friday we'll start unit 2 because we're kind of doing our review today. Unit 2 will start with chapter 5 on Friday. If you haven't taken your exam by then, that's fine because it's not due till midnight on Friday. But we're going to start our lecture and see how far we get on chapter 5. I'll try to slow down so we have more time. Okay, so for chapter 1, where we're talking about the properties of life, and we talked about the scientific method, and the steps in the scientific method. And I'm just looking through my book as I'm talking here. And we talked about peer review, talked about science versus pseudoscience, graphs and tables, and then those major themes that are running throughout, like evolution and structure and function, energy and matter pathways, etc. So going through chapter 1, are there any questions that you're not clear on? Or if you think if you have questions about the exam, about chapter 1, anything about chapter 1, please feel free to speak up. Okay, so nobody's saying anything. Okay, chapter 1 was a nice introductory chapter. Chapter 2 is the chemistry chapter. I know I had questions about ionic bonding, so I'm going to go over that. After I go over that, if you have any other questions about the chemistry chapter, please let me know. I mean, go look through your notes, look through your book. If you have questions, please unmute yourself and let me know. Okay, so I am going to pull up my PowerPoint for chapter 2. I think I have to share my screen. I can't share screen. Chapter 2. Okay, so can you see the PowerPoint now? Anybody? Yeah? Okay. Cool. So, question? Okay, so I'm going to go, we're going to talk about, oh, this one I have drawings in there. Sorry about that. We're going to look, so salt is a good example of an ionic compound. And this is because, to have a periodic table. Okay. Oh, sorry, it is drawn all over. So in the periodic table, we read it from left to right. And we are able, so if I can,
So
there we
have the periodic table.
What?
So we have,
Sorry, I don't know why it's talking. I'm so sorry. Okay, so the periodic table, we read it from left to right. On the left, right here, you're going to find ions because they're going to be most stable when their electron shells are full. And the most stable elements that have all of their electrons are the noble gases. So they're very, very stable. They have all of their electrons. But so if we go from helium and we go to lithium, which is 3, helium is 2, lithium is more stable when it gives off an electron. So it carries a positive charge. So lithium has a positive 1 next to it because it gave up an electron to make it more stable. And on the other side, so if we continue on here, oh, wow, that changed things. If we look here at fluoride, that's number 9. That is more stable when it has an extra electron because then it has its full electron shell. And so you'll find that often elements in this row right here carry a negative charge because they accepted an extra electron. So that's kind of the background for ions, and I'm going to find the ionic bonding section here and go through that more. Let's see. So with ionic bonding, because we have atoms that are more stable giving up an electron, so they'll carry a positive charge, or they're more stable accepting an extra electron, so they would carry a negative charge. So ions themselves carry a charge, whether it's positive or negative. It is a type of bonding. So here we have our slide here for ionic bonds. So if we look at sodium chloride, we look at salt again. Sodium is on the far left of the periodic table, so it's more stable, giving off an electron. so it will carry a positive charge. Chlorine, on the other hand, is on the far right of the periodic table, and it's more stable accepting electron. So it carries a negative charge. When you have a positive charged ion and a negative charged ion, they're going to attract to each other. It's like the positive and negative ends of magnets. They're just going to attract to each other. The sodium, the electron from sodium is given over to chlorine, so it becomes chloride. And so if you pulled those apart, they'd have charges, but together, that ionic bond, it holds them together because of that charge difference. So they're attracted together and held together. Let's see. Are there any questions about that? So that made more sense about ionic bonds? Okay, cool. Well, I'm just going to look through chapter 2 and see if there's anything I think that might be helpful for you guys. Was everybody clear on the atom and protons and neutrons and electrons? So for example, if I gave you how many protons there were in the nucleus, could you tell me how many neutrons were in a nucleus? Because that's kind of, you're going to have a couple questions like that on your exam. So let me scroll back here real quick. Okay, so protons, neutrons, and electrons. Quick overview, just a refresher. So protons, that's your atomic number if you're looking at the periodic table. So you need to know your definitions, what an atomic number is, what your atomic mass is. Your atomic mass is found on the bottom of that little square, and that is your protons plus your neutrons. So if I gave you the atomic number and the atomic mass, for example, could you tell me how many neutrons were in an atom? You're just doing some simple subtraction or addition. So can I draw on this? So if our atomic mass was 14 and our atomic number was 7, 14 minus 7 is 7, so that would give us 7 neutrons. This is not in an isotope. This is just regular, straight up atom. So you would just have to do some simple subtraction or addition to determine your atomic number, your atomic mass, or how many neutrons there are. There are a couple questions like that on the exam, so you're going to have to make sure you remember how to kind of convert those things. Let's see. Were the other types of bonding clear? So covalent, polar covalent, and non-polar covalent bonds. I see people nodding their heads. Okay, good. Okay, if I give you a pH, let's say I give you a pH of 8, can you tell me if that's an acid or a base? Anybody?
It's a base, right?
That is a base, correct. So you'll have a couple questions that are asking you about the pH scale, identifying whether it's an acid. So an acid is between 0 to 7, and a base is from 7 to 14. Completely neutral is at 7. So you need to know your basic acid to base range. Okay, are we clear on dehydration synthesis and, oh, what's the other one? I just blanked. Hydrolysis. So do I need to go over that, guys? I'm going to go over it real quick because, I'm sorry? I'm going to go over it real quick because some people tend to get confused between those. Oh, I think I asked a question about ocean acidification. Do you guys remember this slide? So quick recap on ocean acidification is that extra carbon dioxide in the atmosphere is absorbed into the salt water, which then lowers the pH in the ocean, which makes it hard for animals to make shells or coral to build coral. So it reduces their ability to build their skeletons, essentially. So extra carbon dioxide in the atmosphere is the reason you have ocean acidification. Which, just a side note, since this whole pandemic has occurred, the pollution levels across the world have gone down dramatically. It is amazing. It's awesome to see places where there are pollution constantly suddenly have clear blue skies, like Los Angeles, because there's so many people who are not driving, who are working from home. It's amazing the difference we can see in just 2 months of reducing our carbon footprint. Just a little side note. Okay, so let's get to our chemical reactions. Okay, hydrolysis. Hydrolysis, if we look at the word, hydro refers to water, lysis refers to bursting or breaking. So try to remember, in this type of reaction, hydrolysis is using water, it's breaking water. So that's literally what hydrolysis means, is bursted water. So it's breaking the bonds in water to separate, to break the bonds in a compound. So using water to split those two molecules apart. So think about lysis. The whole word lysis kind of refers to burst, destruction. Okay, so we are destroying that chemical bond. We are breaking that chemical bond. So if you can kind of keep that idea of lysis, breaking a chemical bond, breaking water, that should help you. So anytime you see a reaction where water goes in and you're splitting up a molecule, that's going to be hydrolysis. Dehydration synthesis is the opposite. So dehydration, you're losing water, just like dehydration sounds like, and then synthesis is building. So this is where you are creating chemical bonds, building a polymer from monomers. And so it is the exact opposite. And you lose a molecule of water when you do this reaction. So dehydration synthesis, is shown here, is the exact opposite of hydrolysis. So try to just take that word apart, and you should be able to deduce what's going on. If I ask you a question about dehydration synthesis or hydrolysis, just take the word apart and you should be able to figure it out. I'm gonna keep looking here. Fats. There are different types of fats, right? Yes, yeah. There are different types of fats. Question, are all fats bad for you? I see some people shaking their head. That's right. So there's good fat. There's good, did I get another person in here? It serves purposes within the body. So will anybody be willing to kind of share what a purpose of fat is in the body? Because this is a question on your exam. What are good fats? What are purposes? I'll find that slide for you guys. I want you to answer though. Okay, so here's the slide talking about fats. So fat is not necessarily a bad thing. In excess, yes, but it has essential functions. And so here, this shows you the essential functions: energy storage. So that's where we have that adipose tissue storing the extra fat. Cushioning, you have fat that surrounds organs to help protect them. Insulation, so helps keep your organs warm so they can function properly. You have fats in all your cell membranes, so phospholipids is a fat. Cholesterol is also found in that plasma membrane, helps keep the fluidity of that plasma membrane. And then you also have steroid hormones, which are based off of fats. So these are good functions of fat. So not all fat is bad. So that it serves purposes. Okay, see what's next. Oh, okay. What kind of bond holds amino acids together? See, this will be on page 38 if you want to look real quick. Nobody? Okay, the type of bond, you'll have to know this, is a peptide bond. Okay? And it's specific to joining amino acids. So amino acids are held together via peptide bonds. Remember that. Okay. How do we feel about enzymes? Any questions about enzymes?
Can you go over that just a little bit more?
Yeah, sure. Let me find those slides so you have some illustrations to go with it. Is that it? Nope. It's way down at the bottom. Okay. So enzymes are proteins. So remember that they are proteins. The shape of a protein determines its function. So specific shape is very important. There are enzymes that help lower the activation energy of chemical reactions. So they make it easier for a chemical reaction to start happening in a cell, in a body, okay? Let's see. So sometimes a lot of chemical reactions can cost a lot of energy to get started. So if you have an enzyme that can help lower how much energy is necessary to start that reaction, then that reaction can start and keep going. So all of those chemical reactions that are going on inside your body pretty much all need enzymes to help them go. So I'm not going to ask really detailed questions about enzymes. One of the key things that you need to remember is they lower the activation energy. So they reduce how much energy it's going to take to get that reaction started. How they do that is by binding to a substrate. So in this example here, we're talking about the lactase enzyme, which helps break down lactose, that sugar in milk. What that does is when lactose binds to the active site of the enzyme, it changes the shape of the enzyme. And that changing of the shape, you have a nice tight fit, allows that lactose to be broken down. So this enzyme breaks down lactose. Without this enzyme, or an enzyme that maybe has a mutation, so the shape is just not right, then you won't have that chemical reaction occur. So you can't do it without the enzyme. Having a functional enzyme is key. Let's see, do I have pictures? Okay. So it doesn't actually take energy to do this. So just remember that, but it lowers the overall energy to do the reaction. So there are inhibitors they're competitive inhibitors and non-competitive inhibitors. And so what can happen is you have your enzyme and you have a substrate that binds to the active site. In this case, we're looking here, a competitive inhibitor. Sorry, my phone's ringing and it's driving me nuts. Okay. So the competitive inhibitor will bind to the active site, and by doing that, it blocks the actual substrate from binding. If it blocks the substrate from binding to it, then the substrate you can't have a chemical reaction occur. So this is an inhibitor that inhibits the reaction by blocking the active site. Okay. In a non-competitive inhibitor, you have this non-competitive inhibitor bind to a different site on this on the enzyme. And when it binds to that site on the enzyme, it changes the shape. And remember, the shape of the enzyme, the shape of the protein is important in how it functions. If you change the shape, it's not going to be able to function properly. So in this case, if we change the shape, the substrate can't bind to the active site because the shape isn't right. They have to be like a puzzle fit. So because the shape changed, the substrate cannot bind, and then the chemical reaction will not occur. So this is one way your body prevents chemical reactions from occurring, is by putting out competitive or non-competitive inhibitors that blocks the enzyme from functioning. Without those inhibitors, the enzyme will be able to function. It's easily recycled, so it goes over and over and over again until there is no more substrate, or until an inhibitor is present and it won't be able to work anymore and carry out that chemical reaction. Did that help? I see, okay. Any other questions about that? Do I need to clarify any further? So people are saying okay. Okay, so we're not going in depth. You just need to know the basics of what an enzyme is. Structure determines the function, what kind of inhibitors we have, and the fact that an enzyme lowers the activation energy. Okay, that's a key kind of phrase that you will need to know. Okay, so then that's the end of chapter 2, and we're moving into chapter 3. So I'm going to close this and, oh, okay, screen sharing. All right, chapter 3. So before I get started, are there any questions related to chapter 3? So this is the cell, the structures with plant cell, animal cell, plasma membrane. Oh, diffusion and osmosis, passive transport, active transport. Let's see. And then we have kind of the basics of the different types of organelles and structures within a cell. Okay, I'm guessing that one place we may have questions is going to be with diffusion and osmosis. How are we feeling about that? So, okay. I'll do a brief overview then if there aren't specific questions about it, and if you have questions that come up, then just speak up, okay? Plasma membrane is made up of phospholipids, so remember our phospholipids, and it's a phospholipid bilayer, so 2 layers. It's like a sandwich where you have a phospholipid head, tail, and then another phospholipid that's backwards where you have the tail and then the head. So you have a sandwich, or the heads are the bread, and the tails are the filling of your sandwich, which now makes me want to have a sandwich. So talking about transport methods across that plasma membrane, I am just going to go to the slides. Let's see. So here's a reminder of phospholipids, phospholipid membrane. So, move that. Okay. There are different types of transport. So I'm going to go over the types of transport and the types of diffusion. There will be questions about diffusion and osmosis, so you do need to know the basics of those. Okay, so the background is that we have a plasma membrane. Every membrane is selectively permeable, which means it lets some substances across and prevents other substances from passing through. So it carefully regulates what goes in and out of the cell. Okay, so concentration gradient. How do we feel about concentration gradients? Anybody? Okay. So with a concentration gradient, if we look at this picture here, we have a beaker of water and they're dripping in dye. So let's look at just the concentration gradient. When you first drip in dye, we have a concentration gradient where we have a lot of dye molecules in one spot. But because atoms tend to move around and jostle each other, this is going to cause them to bounce off each other and eventually spread out to fill up the space in that beaker. That's a flask, sorry, in that flask. And that eventually mixes so much that you have an equal concentration gradient. So at the beginning here, where we have all of the dye in kind of one spot, we have a concentration gradient where we have a high level right there where the dye is, so the high concentration, and then a low concentration where it's just pure water. What happens is molecules are going to tend to move from areas of high concentration down to areas of low concentration. And that ultimately ends up where you have those molecules that reach equilibrium in concentration. Okay, different types of transport. You have passive transport, and this does not require any energy. And so we're going to talk about the different types of passive transport. But what happens here is substances move with or down their concentration gradient from areas of high concentration down to areas of low concentration. Active transport is the opposite. It requires energy, and you're basically pumping molecules against their concentration gradient. You're making that concentration gradient even bigger. So if you were able to reconcentrate all those dye molecules in one spot, that would take energy, and it's moving them against their concentration gradient because naturally those molecules are wanting to separate and spread out to fill up the space that they are in. So, talking about passive transport, diffusion. This is the simple movement of molecules from areas of high concentration down to areas of low concentration. It does not cost any energy. There's no ATP that's expended. And these are typically moving down through a plasma membrane. If we're talking about plasma membranes here. If we look down here at the bottom illustration, it kind of shows you, if we were just talking about dye molecules in a bottle here with a plasma membrane there. Over time, so we have a high concentration gradient here, and those molecules are going to bounce off each other and move with random motions until they eventually spread out to the other side and become equalized in concentration. Okay. Do we need to talk about the stuff inside the cell? I don't think so. Okay. Passive transport of water. This has a specific name, so you need to know the specific name is diffusion. The specific name of diffusion of water is osmosis. Okay. So diffusion is any other molecule, but if we're talking about diffusion of water, that is called osmosis. So this happens when substances are not able to pass across the plasma membrane, but water is. And so it's the same principle. You have a high concentration of one side and a low concentration on the other. So if we look at this illustration right here, you have a high concentration of sugar on one side and a very low concentration on the other. The sugar cannot pass through that membrane, but water can. And so, because it still is trying to equalize the concentrations on both sides, remember it's random, but the ultimate end goal is to equalize those concentrations. So because the molecules can't move across the plasma membrane, water moves across the plasma membrane, and it causes a really cool phenomenon where the water level on the other side of the plasma membrane increases. So if you did this in a beaker like this, where you were able to split it with a membrane, you would see the water rise on the side that had a higher sugar concentration. And the water would continue to flow, ideally, until the sugar concentration on one side is equal to the sugar concentration on the other. So this is just the diffusion of water across the membrane. It still is trying to reach that equilibrium, but it kind of does it in a little bit of a different way. Are there any questions about osmosis? Okay. Another type of diffusion, facilitated diffusion. This again is free. It doesn't take any cellular energy, but it does use a protein, a plasma membrane protein. So there are lots of proteins embedded in the plasma membrane. Some are transport molecules. So in the case of facilitated diffusion, you still have an area of high concentration and an area of low concentration. The molecules are still moving from areas of high to areas of low, but they can't pass through that plasma membrane without the help of a protein, a carrier protein. And so if you look at the illustration down here below, basically the protein molecule, you have a molecule binds to that transport protein, causes a shape change in the transport protein, which then allows the molecule to enter the cell. And then it goes back to its normal shape, and then it can accept another molecule binding to the cell. So that's the only way these molecules are able to enter into the cell is through this transport protein. Active transport requires energy in the form of ATP. And this, again, this is where you're moving substances from areas of low concentration up to areas of high concentration against their concentration gradient. So essentially, you're building up the concentration and making it higher, that concentration difference, by using active transport. So in this illustration, and this is on page 49, this is showing a sodium-potassium pump. And sodium-potassium pumps, they're pumping out 3 sodium ions for every 2 potassium that come in. And that takes ATP to do that. What's the whole point of the sodium-potassium pump? It's to increase the concentration gradient across the cell. So you have more positive sodium ions outside the cell than you have inside. So you would have a net negative charge outside the cell. So that's the purpose of the sodium-potassium pump. But the point is that it takes energy. It's building up the concentration gradient, making it higher. And you can only do that when you use energy. Okay, let's see. Endocytosis and exocytosis are pretty explanatory. This is a nice summary of the different types of cellular transport. So simple diffusion through a phospholipid bilayer. So this is just going down its concentration gradient. Facilitated diffusion uses a transport protein. Active transport requires ATP, and this is where you're building up your concentration gradient. You're making it bigger. And then this picture here is osmosis, illustrating what happens with the water flowing. So remember, osmosis is diffusion of water. Okay, let's see. Are there any questions about the organelles or their functions? We feel good about that? Okay. I will ask you, like on the exam, there are questions about what's the difference between a plant cell and an animal cell. You should know what structures are unique to plants and what structures you only would find in animals, because there is a question or two about that. Okay, then that takes us to chapter 4. So let me close this and pull up chapter 4 real quick. Okay, so we got chapter 4 here. Come on, big screen, you can do it. There we go. Oh, has all my notes on there. Sorry about that. Okay, so chapter 4. We'll skip that slide because it's very busy. There are definitions that you'll need to know. So if you know your definitions, it's going to help you answer some questions like what is energy? What is work? So work, there's your definition. So your definition for energy is defined as the capacity to do work. Work is a force acting on something to make it move. So if you think about it, anytime you touch something, even if I don't move the pen, I'm exerting force. So work is force that actually causes it to move. So that I just did work. The difference between kinetic energy and potential energy. Do we have questions about that? We feel good about it? Anybody? That's cool. Okay, so I won't go over that. ATP, super important that we understand ATP. That has 3 phosphates. That high energy bond is at the last phosphate bond. When you break that bond, it gives off energy in the form, it gives off energy so that you can do work. Because a high energy chemical bond. ATP is constantly recycled. It's reused and rebuilt throughout the different chemical reactions. So make sure we know that. Understand the principles of energy conversion. So we absorb energy in different ways, but we also give off energy. Important to remember the principle of conservation of energy, that energy is never created or destroyed, but it can be converted from one form of energy to another. Okay, let's see. Okay, entropy. We need to remember what entropy is. So the entropy is how much disorder there is in a system. Things continually work to increase entropy in the system. However, we have this question here, which is still an important question. Why don't living systems like ecosystems or organisms fall into complete disorder due to entropy? Because if we follow that law, the second law of conservation of energy, that things go to increase entropy, then why don't we see more disorder in a system? Guys, any ideas? Okay, that's something I want you to think about. Maybe I'll make this your question for your attendance today. I'm going to do that right now. Whoops. June attendance. Someone want to read it to me real quick? Make you talk. No?
What do you want to read?
The highlighted question. I'm going to type it into your Zoom attendance.
Why don't living systems, comma, like ecosystems or organisms, comma, fall into complete disorder?
Ecosystems and organisms. So why don't living systems like ecosystem and organisms I can't spell it, organisms fall into complete sorry, 1 second. So I am making that your attendance question for the day. So make sure after our lecture today or sometime before the end of the day, you go in and answer that question. It doesn't have to be right, but I just want a thoughtful response, okay? So I will go back to this. Okay. Differences between producers and consumers, is that pretty clear? How do you feel about that? That part's pretty cool. We are all, what are we? Are we consumers or producers?
Consumers.
Yes, thank you. I appreciate that. We are consumers. Plants, on the other hand, are producers. We eat the producers. Okay. So plants get their energy from the sun. And I know this is all written up on there from our lecture. That is okay. So solar energy is inputted on a daily basis, and energy is going to flow from solar energy into the plant, into the system. And that's why we say that the energy flows through the system, because it's going from different parts of the system to another. So what you need to know about photosynthesis, you need to know the overall reaction, and we'll come up with that a little bit later as well. But knowing the chemical reaction for photosynthesis and the chemical reaction for cellular respiration, know both of those. It's pretty easy because if you know one, you know the other because they're opposites of each other. So it's pretty helpful in that way. So producers, consumers. Okay, here is our overall equation for photosynthesis. Carbon dioxide, where do we get the carbon dioxide? Do you remember? The carbon dioxide is from the atmosphere. It enters through the leaves. On the underside of leaves are these little pores called stoma, and they're like little mouths. And it breathes in the carbon dioxide, and that's where you get the carbon dioxide to run photosynthesis. Water comes from the roots, and then we have our sunlight. You are able to build sugar from that carbon dioxide, and you have a waste product of oxygen. That oxygen leaves through the stoma of the leaves and enters back into the atmosphere. Okay. Photosynthesis, I had a question about the thylakoid membrane, so I'll talk about that when it comes up. But photosynthesis takes place inside the chloroplast. This picture here is of an illustration of one chloroplast. Okay, let's see. All right. So photosynthesis is done by plants, by producers, convert solar energy into chemical energy. That chemical energy is sugar. Cellular respiration, on the other hand, both producers and consumers go through this process, and it's the only way to make ATP, essentially. So plants have to go through photosynthesis, and then they go into cellular respiration. So plants have both chloroplast and mitochondria. They do both. Animal cells only do cellular respiration. That's a question you may see. Okay, let's see. You don't need to worry too much about NAD or FADH, okay? Just the basic thing you need to remember is it carries electrons and it carries hydrogens. Those are used in some of the different reactions later on in photosynthesis and cellular respiration. This is just the recycling of ATP and NAD. Let's see. Trying to get a little bit closer to that question about thylakoid membranes. Okay, looking closer at the chloroplast. made up of a lot of little membranes stacked. They look like little pancakes, or if you cut them in half, they look like stacked pita bread. Each of those stacks, each one of those round structures is called a thylakoid. They're like little discs. When you have a stack of thylakoids, that's called a granum, or grana is singular. And so this is where you have photosynthesis occurring, is across that thylakoid membrane, or part of it's occurring across the thylakoid membrane. Okay, I'm not going to ask anything about chloroplast. Okay, I know I'm skipping through some stuff. If you see something you have a question on, please speak up. The difference between light reactions and dark reactions, I do believe there is a question about that. I'm pretty sure. Okay, so light reactions require sunlight to work. The Calvin cycle, or dark reactions, or the light independent reactions, this is where you're making the sugar. So you have light reactions that require sunlight, and then you have the Calvin cycle, or the dark reactions that do not require sunlight to happen. So plants can actually do this part at night. They don't need the sun to do it. Okay, so how does this happen? So energy from the sunlight is absorbed by the chlorophyll pigments in the thylakoid. Okay, this first step, this is a light reaction. Water that you get from the roots, water is split, releasing oxygen, that's our byproduct, and then you have extra hydrogen ions, which are used later. So in this first part of the light reaction, you produce a little bit of ATP and a little bit of NADPH. Those two things are used later on in the This first part, where the sunlight hits those chlorophyll molecules, this takes place on the thylakoid membrane, because those little pigment molecules are embedded in the thylakoid membrane. So the light hits those pigment molecules, you have the splitting of water, then you have oxygen as a byproduct, and you produce ATP and NADPH. This is just, oh, that's probably confusing. We don't need to talk about that part. Okay, so the light reaction is really simple, happens across the thylakoid membrane. Then we have the Calvin cycle. Let's find Calvin cycle. Calvin cycle is where you use the carbon dioxide and you produce sugar. And this is light independent reaction. So we already asked the question of where do we get the carbon dioxide? From the stoma of the leaves of the plant. Okay, Calvin cycle, where does it take place? It takes place in the stroma. So the stroma is the fluid surrounding the chloroplast. So light reactions take place across the thylakoid membrane. Calvin cycle takes place in the stroma. So knowing locations is good. Trying to see what else we need to worry about. Okay, looking a little closer at the light reactions, this is showing you the individual pigment molecules. So we have, there's our little pigment molecules. The sunlight hits it, and that causes the water to be split. The main pigment molecule that we'll ever talk about is chlorophyll. All right, do you remember this slide? Yes, no, maybe so. Okay, this is also found on page 67. And so this breaks the light reactions down even further. Are there specific questions about this page? Because I will be happy to go over it again, but not if you don't need it. Anybody? So one thing to remember, all light reactions are occurring across the thylakoid membrane, okay? So these are photosystems that are embedded in the thylakoid. The light hits it, we split water, it excites the electrons. Those excited electrons travel down what we would call the electron transport chain, where ATP is made. those excited electrons move to the next photosystem, which excite the electrons even further, and then it creates NADPH. Those two products, ATP and NADPH, are used in the Calvin cycle. So it's kind of setting up the ingredients for what you need to do your Calvin cycle. Okay, we talked about where oxygen goes. So remembering Calvin cycle happens in the stroma. Our carbon dioxide comes from the stomata of the leaves of the plant. And the whole purpose of the Calvin cycle is to take those carbon dioxides and make glucose or other compounds with it. Glucose being the main thing, sugar, C6H12O6. And then the plant can use those sugars in different ways. It is also able to produce other compounds. So let me find that. I'm trying to find that picture. So if you look at page 76, you know that, oh, that's cellular respiration. I'm getting ahead of myself. Sorry. Never mind. Okay, Calvin cycle. What goes into the Calvin cycle? So the ingredients, ATP and NADPH, we got that from our light reactions. And then carbon dioxide we get from the air. The outputs of the Calvin cycle produces a 3-carbon sugar. Plants use that 3-carbon sugar to make glucose or other compounds. And then if glucose isn't used right away, it can be stored. And if you want to make sugar, the Calvin cycle has to go through 6 cycles to make 1 glucose molecule. Okay, plants use sugars in many different ways, and how they're constructed kind of determines their function. So in this picture, it just shows you 2 examples. We have starch and cellulose, which are 2 ways that plants store sugar. Okay, any questions? Because I know we had questions on thylakoid membrane. How are we feeling about cellular respiration? I see somebody, Sydney's nodding her head. Everybody else, I want to see your video, seriously. I had to like put a nice shirt on and do my hair for you. All right, I'm just teasing. Okay, I'll look through here and see if there's anything I think is important to refresh your memory on. So one thing I did mention earlier is that all organisms, both plants and animals, so producers and consumers, carry out cellular respiration. So plants carry out photosynthesis and cellular respiration. Cellular respiration is aerobic, it requires oxygen, takes place in your mitochondria. Let's see. This is just, you guys know this part. Okay, body systems that play a role in cellular respiration. There's 3. I put that wrong. We have respiratory system, circulatory system, and digestive system are all involved in cellular respiration. Okay, let's see. We don't need to worry about that. Okay, so where photosynthesis can occur in different stages at different parts of the day, cellular respiration happens 24 hours a day on a constant basis in every cell in the body. And that oxygen that we breathe goes directly to cellular respiration, and we need it on a constant basis. Okay, so byproducts of cellular respiration: carbon dioxide, which we breathe out, water, which we use in our body, and energy. The main production of energy happens in cellular respiration. So this illustration here is just showing you, this is on page 72 and 73, the overall picture of what's going on with cellular respiration. And there's 3 major stages. You have glycolysis, citric acid cycle, and electron transport chain. So photosynthesis and cellular respiration both have electron transport chains. Okay, so that kind of helps with understanding them a little bit. Okay, so we'll go briefly through each stage. You don't need to know a lot of details because this is just general biology, but you need to know the basics. Okay, so one of the things you definitely need to know is the chemical equation for cellular respiration. And remember that it is the opposite of photosynthesis. So this picture is just illustrating how photosynthesis and cellular respiration are very closely related. Okay, 3 major stages. You should know that there are 3 stages to cellular respiration. You have glycolysis, citric acid cycle, and the electron transport chain. So in glycolysis, you're taking glucose and you're splitting it into smaller subunits. So it's broken down into 2 pyruvic acids, which are 3 carbon chained molecules, and you produce a tiny amount of ATP, and you produce a little bit of NADPH. Those are going to be used later. This is not the main purpose of glycolysis. The main purpose of glycolysis is to break that 6 carbon sugar down into smaller subunits. Then the next step, this is in the citric acid cycle, you take that 3 carbon pyruvic acid and you split it into acetyl coenzyme A, that's acetyl CoA is acetyl coenzyme A. Oops, what happened? Sorry, my slide just got big. Okay, so you split it into that and to carbon dioxide. That carbon dioxide is a waste product, you breathe it out. That's where we get our carbon dioxide. It comes from the citric acid cycle. This portion of cellular respiration takes place in the fluid of the mitochondria. And what happens in the citric acid cycle, it continues to break down acetyl coenzyme A into smaller subunits until they're all broken down into carbon dioxide and exhaled. You produce a lot of NADH and FADH, and those will be used in the electron transport chain. And a small amount of ATP is used. But the major player that produces the most amount of ATP is the electron transport chain. This takes place across that inner folded membrane of the mitochondria. And what happens is you have NADH helps excite these electron carriers, pumps hydrogen ions out of the cell, builds up the concentration gradient of hydrogen ions. So this is using energy. So it's using FADH and NADH to pump out these hydrogen ions to build up a concentration gradient. The whole purpose of building up that concentration gradient. Oh, I'm sorry, I'm trying to increase the size of this slide. Nope, not going to work. Okay, so the whole purpose of building up this concentration gradient is so that those hydrogen ions will flow through that transport protein and produce ATP. It's kind of like a wheel or like a fan. And the hydrogen ions are flowing through and it causes it to spin, and that produces a lot of ATP energy. So it's kind of one way to visualize it. Let's see, are there any questions about the 3 stages? So we have glycolysis, citric acid cycle, and electron transport chain. Those are the 3 main things that are happening in cellular respiration. You should know where they occur and which step produces the most amount of energy in ATP. Okay, I don't know if I'm going to ask anything about carbonation. Okay, I will ask about anaerobic respiration. And so this is where you are carrying out cellular respiration without oxygen. So anaerobic, AN is referring to without oxygen. And so it goes through it goes through the glycolysis just fine. It breaks it down into pyruvic acids, but then it can't do the rest of the steps. It has to go through anaerobic fermentation instead. And that produces different byproducts, and it produces a tiny bit of energy, but not very much. So fermentation is anaerobic. and it does not produce very much ATP. Important to remember. Lactic acid is when you have fermentation in your muscles occur and you don't have enough oxygen. Lactic acid fermentation is kind of an emergency mode to make energy. It's not going to make very much. It's going to make your muscles burn and it causes muscle fatigue, but it can get you through a little bit of extra energy expenditure before you completely run out of energy. Okay, to release. Check our time. Oh, we're doing pretty good. Okay. Just a quick reminder. This is, you can see it on page 76. Cellular respiration does not just break down sugar. Okay, that's the main thing it does, but it also can break down fats, carbohydrates, and proteins. And so that's important to remember. It may produce a tiny bit of energy from those. They're not the main energy producers, but it can process those other types of molecules. So you can see those here. And so the ATP that you're producing through cellular respiration basically helps run all of the processes that are occurring in your body right now, which is kind of cool to think about. These reactions are occurring on a constant, steady basis, a lot, very fast, and that's providing you the energy to think. Your brain is functioning, keeps your eyes open, all the energy you're expending, even just your metabolism, the reactions that are occurring in your body to keep you functioning, those take energy in the form of ATP. Okay, whoops. That is the end of chapter 4. Are there any questions on chapter 4 that I can address? Sydney says no. Anybody else? Okay. So we'll end a little bit early then. Let me figure out how to close this. We have another Zoom on Friday. So let's see. So another Zoom on Friday. So we'll probably start chapter 5 on Friday. Your exam is Friday. Got that fixed. And okay, we'll start chapter 5 unless there are more questions. If there are more questions, please email them to me. I will address them before I start chapter 5 on Friday. Your new stuff for unit 2 will become available after your exam on Friday. So you have some time. So please remember, homework is due on Friday. Labs are due on Friday if you're in lab. If you're having trouble with Labster, please let me know. You do need to get a code from the bookstore to access Labster, and it should be all set up. If you get a code and you're still having trouble, please let me know.
Can we just call the bookstore and then they give you the access code, or?
You have to pay for it. So what I was told is that they will email it to you directly. So it's really quick. So if you haven't done it yet, I would do it ASAP because labs are due by Friday. So you'll be busy. Yeah. Okay, any other questions?
Would chapter 5 be part of unit 2 exam?
Unit 2, yes.
Okay.
Good question.
I tried to get into quiz for chapter 4 and it said it's not open until like Saturday, I believe.
Yes, it's not going to be open until we finish this unit. And then it will be open the next day. Are you just wanting to work ahead?
No, the quiz for chapter 4. I thought that would be in this one.
Oh, quiz for chapter 4. Okay, that's quiz 3. And let me double check that right now while I'm on the phone with, not on the phone, while I'm Zooming with you. So quiz 3. Okay, it should be available. I know it wasn't available earlier. There was a problem with it. I'll double check the dates. Okay, it does show June 3 in here. Let me save it. Was this recently that you couldn't access it?
Oh, I thought quiz 4 was for chapter 4. That's just my bad then.
Oh, okay, okay. No, quiz 3 is for chapter 4. Quiz 4 is the next unit. The numbers they don't match up completely because I don't have a quiz for every single chapter. Okay. Any other questions, concerns? Okay. If not, then I'm gonna let you ladies and gentlemen go and see you on Friday in Zoom again. So we have 3 Zooms this week. Please remember to do your little question for attendance so that you get credit for being here on Zoom today. I want to make sure you get points. And in the meantime, if there are questions, you can text me, email me. During the day, I won't be able to answer my phone if you call. I'll be able to answer after about 4:30. So just keep that in mind. I can answer a text pretty quick, but I can't take a lot of phone calls because I have my other job I'm working. All right, I will let you go. Thank you so much for joining in Zoom today, and I will see you all on Friday. Bye.